Welcome to the BioWhisperer channel. Our topic today is on a perspective overview of fishmeal cell culture. If you are new here, we are a group of retired professors sharing bite-sized videos in life sciences. Let's dive into the topic for today. Fish cell culture. The overfishing of oceans and the impact of climate change have led to significant declines in wild fish populations, and the reliance on fishmeal in aquaculture may not be a sustainable solution to feeding the world's growing population. The production of seafood from fish cell and tissue cultures, a form of cellular agriculture, offers a potential alternative that could address these sustainability issues. While the concept of producing meat from cell cultures has been widely discussed, the application of cellular agriculture to the production of seafood has not been fully explored. There are scientific and human factors, such as cultural and regulatory considerations, that must be taken into account in the development of cell-based seafood, but the potential for preserving marine environments makes this pursuit especially important. Cellular agriculture involves the production of agricultural, or farmed, products using cell cultures rather than entire plants or animals. There is increasing interest in this field as a way to address issues such as public health, animal welfare, and the environment that are associated with traditional animal agriculture. One application of cellular agriculture that is being explored is the creation of seafood using fish cell and tissue cultures. This approach, known as cell-based seafood, combines advances in biomedical engineering with modern aquaculture techniques. Biomedical engineering techniques, like closed-system bioreactor production, can be used to produce marine animal cells on a large scale. In addition, advances in aquaculture techniques, such as genetic modification and closed-system production, can be applied to cell-based seafood production. The use of seafood waste products, like chitosan, a derivative of mushrooms, as scaffolding for crustacean tissue, may also be promising for cell-based seafood production in bioreactors. However, further research is needed to understand fish muscle cell and tissue cultivation, develop serum-free media formulations optimized for fish cells, and design bioreactors specifically for fish cells to enable large-scale production of cell-based seafood. The existing challenges with aquaculture. Aquaculture has surpassed marine capture as the primary source of seafood for human consumption. Global landings for marine capture have remained stable at around 90 million tons per year since 1994, while global aquaculture of fish and shellfish has nearly doubled over the same period. However, many farmed fish depend on feed made from wild-caught fish, meaning that aquaculture may actually be decreasing the global fish supply rather than increasing it. While controlled aquaculture environments are more efficient production systems than using natural conditions, there is currently no research on the use of fully controlled environments for producing seafood from cell cultures rather than marine animal husbandry. Aquaculture, the farming of aquatic organisms, has a long history dating back to ancient Egypt. However, it was not until the 1970s that aquaculture became a significant part of global food production. As with the intensification of land animal farming, the growth of aquaculture has led to increased control over growing conditions through methods such as feeding, breeding, disease control, and waste removal, resulting in higher production rates. However, this intensification also carries risks, such as the production of excess nitrogen and phosphorus waste, which can harm waterways and contribute to algal blooms. There is also a risk of pathogen transmission from farmed fish to wild species. Additionally, the lack of research on the welfare of most aquaculture species has made it difficult to establish appropriate regulations and standards for the ethical treatment of farmed aquatic animals. Genetic modification has led to significant improvements in the production, efficiency, and development of fish in controlled aquaculture systems. For example, genetically modified rohu grow 4 times faster than non-modified fish, and genetically modified mud loaches and tilapia can grow much larger and faster than their non-modified counterparts. The use of genetically modified fish, such as aquabounty salmon, which grow twice as fast as wild salmon and have a higher feed conversion rate, has been approved for sale in Canada. However, regulatory and legislative barriers have prevented the sale of these fish in the United States. These advances in engineering biology for closed aquaculture systems may provide a foundation for future research and development in cell-based fish production. Current research progress. There has been a significant increase in research on cell-based meat production, including the development of startups in this field, updates to life cycle assessments, and publications focus on refining the technologies required for large-scale production. However, the selection of cell species and types for cell-based meat production has been largely driven by market size and environmental impact rather than the suitability of these cells for bioreactor cultivation. There has also been growing interest in the regulation of cell-based meat products in the United States, with the Food and Drug Administration and the United States Department of Agriculture both potentially having jurisdiction over these products. In contrast, cell-based seafood production, which has the potential to address the demand for seafood while avoiding the challenges of industrial aquaculture, has received relatively little attention from a regulatory standpoint. The production of cell-based seafood involves the optimization of cell lines, media formulation, and bioreactor design, as well as the use of biocompatible scaffolds for the growth of three-dimensional tissues. While a muscle protein production system for goldfish was developed in 2002, there have been few other published examples of cell-based seafood production. Towards cell-based seafood. Cell-cultured seafood isn't just an idea, it's a reality. Cell-cultured meat, in the form of lab-grown beef or chicken, started the trend. Just like their terrestrial counterparts, cell-cultured seafood will experience either success or failure of cell-grown seafood products. This will ultimately depend on cost and the ability to scale output. Cell-grown seafood may start out more expensive than ocean-dwelling fish, but we remain optimistic that technology will improve and prices will drop. Perspectives ahead. Bioengineering, cell biology, and genetics have made significant progress in the understanding of cell and tissue culture over the past 2 decades, but most of this research has focused on mammalian systems. In contrast, research on the in vitro cultivation of cells and tissues from fish and other marine species has been limited, despite the well-established importance of understanding native fish muscle physiology for aquaculture. Some studies have been conducted on native muscle tissues from freshwater and marine animals, but more research is needed to fully understand the potential of fish muscle in culture.